Well, I'm very pleased to introduce our guest speaker today. This is Dr. Lynn Rothschild from the NASA Ames Research Center. She's a microbiologist, an astrobiologist, and most recently a synthetic biologist. Dr. Rothschild holds degrees from Yale, Indiana, and Brown, and she is currently the chief scientist for synthetic biology at NASA Ames Research Center. She also has affiliations at Stanford and Brown universities. Her research focuses on how life evolved in the context of the physical environment here on Earth and possibly elsewhere. She has flown experiments in high-altitude balloons and rockets to understand the effects of atmospheric conditions on microbes. And her fieldwork has taken her to places I'd like to go if you need a field assistant. She's worked in Mount Everest and the Rift Valley in Kenya, in the Andes, in Yellowstone National Park and New Zealand, where she's studied photosynthesis and DNA damage and repair in microorganisms. And today she's going to tell us about her work in synthetic biology. Thank you.
Great. Thank you very much. In fairness, I have never climbed Mount Everest. I have no intention. But the great thing is you get these astronauts who are a little unlike the rest of us. Their brains are a little different. So Scott Perzinski, when he retired from the astronaut corps, was very kind and brought some samples up Mount Everest for me, and sadly they suffered a tragic fate in the Kathmandu security, from what I can tell, because all the DNA was obliterated. So that's kind of sad. So if anyone here is planning to go to Mount Everest and would like to carry an extra kilogram or so in your future, please let me know. Anyway, thank you very much. I don't know if I'm supposed to look at you or look at you, but I am absolutely stunned by the number of people here listening to me, and thank you very much. You better lock the door before everyone starts to run. As sort of was mentioned earlier, being at NASA, you're sort of expected to reinvent yourself periodically, and it's particularly difficult because you're in an environment with physicists and engineers and geologists and so on. I mean, that's not the hard part. The hard part is that there's no commonality of understanding of the journals. So if I say I had a cover story in the Journal of Psychology or AEM or something, a lot of people here would say, oh, that was great. And my colleagues at NASA would say, what? And if I say to them I had a cover story in Icarus, they'd say, oh, that's great. And you'd say, Icarus? What? So you're sort of expected to reinvent yourself and then get a paper in Nature and, in the next year. And so you have to be somewhat agile on your feet. So what happened is that a couple of years ago, there was a graduate student at Brown University who got very interested in synthetic biology and managed to convince our center director, who is a University of Michigan graduate, that synthetic biology might be a great thing for human space exploration. And next thing we knew, this student had been put into my lab, and I was the supervisor and trying to do a catch-up job very quickly. And so what happened is we went from there to ending up having an iGEM team composed of students from Brown and Stanford, and I'll tell you about their work at the end. That's the part that I'm trying to polish at the end. And so, again, give you an idea of what our current interests are in synthetic biology and where we may be going in the future. Of course, with the election this week, I think that the chances of a lot of what I'm saying today to you will actually happen. I also happen to be the representative for NASA on an OSTP working group out of the White House on synthetic biology. So I think there's a lot of reason to be very optimistic about where we're going on this. So, without further ado, I want to make sure that everyone starts on the same page, and I have to pitch this to all sorts of people. So, the fact is that NASA is an engineering organization. So, if I came to you, if you were an engineer, and I said, look, we've got these things that are programmable, they have built-in solar cells, they're self-replicating, they're modular, they're self-repairing, they have advanced chemical synthesis capabilities, they are exquisitely sensitive at sensing perhaps as little as a single molecule. They can extract minerals with great precision. They can stay dormant with no energy input. You don't need petrochemical products. And they're evolvable. And the really cool thing is that there are about 10 to 20 million variants available on planet Earth. You'd say, give me one now. I don't even want to know what it costs, just give me one now. And of course, what I'm talking about is life. So I'm really glad you're here, Rich, because I swapped that picture out last night. So much for the amoeba. Anyway, the fact is that life has an enormous potential to do a lot of very cool things that engineers would just love to get their hands on. So synthetic biology is a relatively new field that, for the most part, has been started by people who come from an engineering background. In fact, some of them are actually very proud of the fact that they know little to any biology and consider the sort of diversity of life that all of us, I think, in the room and out there in TV land celebrate. And so what they want to do is make it much more tractable to put bits and pieces together and re-engineer. So the party line I hear is, we want to turn biology into sort of these modular blocks that we can screw together. That part of engineering is the idea of having interchangeable parts. So you go down to Home Depot and you get a new part and you screw it together and you know it's going to work. And my answer is the Viking missions and every other mission that NASA's done has not been done with parts from Home Depot. That's not actually part of the definition of engineering. Engineering is doing something useful, making something useful for man. And so this sort of stripped down definition, therefore, of synthetic biology is the design and construction of new biological functions and systems not found in nature. So you will hear some people talk about interchangeable parts. I will at the end, but in fact, it's really just making something new. But the reason that this has really gained traction over more traditional forms of molecular biology is because of the modularity. In addition, of course, the price of sequencing and synthesis has gone down dramatically. So a lot of things that were really bespoke in the past and took, 20 postdoc years to complete, like Jay Kiesling's group making artemisian. It was a real tour de force, but it took an awful lot of manpower. And with these technologies going forward, the amount of manpower needed to make really cool things and very important things very quickly has certainly increased. So the fact is that we are now starting to use synthetic biology on planet Earth to do manufacturing, make pharmaceuticals, materials, biofuels, food, environmental control, and so on. And so the question is, if you're doing all of this, why shouldn't NASA take advantage of this and use these sorts of tools in the same sorts of ways on the moon, Mars, asteroids, and so on? So how can NASA use synthetic biology? NASA and this sort of idea of synthetic biology has come together then in the SynBio initiative. And so what I'll do is give you a little bit of the general sense of how NASA can use synthetic biology and then spend the rest of the time specifically focused on where my heart is, and that's astrobiology. So a lot of people forget that the first A in NASA is aeronautics. We're the National Aeronautics and Space Administration. And so there's certainly a lot of potential for using biofuels for green aviation. People I know are working on it. We're not working on it in-house because I know there's some commercial interest. And so perhaps some of you are interested. The sort of fuels that you then need for these very high-altitude aircraft all the way to space become very different than what you might use on the ground, but it's also definitely a good start. There's been some suggestion of using solar cells. I know there is some sort of trade-off at some point that if you have a massively heavy solar cell on top of your airplane, you may circumvent having to use petrochemical products, but then the weight may be such so that you're ending up having to expend more energy carrying them around in the back of your airplane, so it may not be worthwhile. And I throw modeling up there, as I know there are a lot of modelers in the room, and I've heard whispers that there can be some crossover between aeronautics modeling and metabolic flux modeling, and I don't really know anything more than that, so if any of you do, let me know. So that's sort of my aviation pitch. Then within the National Aeronautics and Space Administration, within the space part, we have a large Earth Science group. I'm officially part of the Earth Science group, and this is not perhaps what you think of. It's not necessarily geology. It's really taking spacecraft, whether it's a low-flying airplane all the way to satellites and beyond, and looking back on the Earth. Now, many of us do field work, and you can be much more sensitive when you do field work in your data manipulation. You can go down to a plot that's a centimeter by centimeter or smaller and know what's going on metabolically. You can know something about the genetics and so on. But there's only so many of these plots you can cover on planet Earth in one day. So if you flip it around, the beauty of using remote sensing is that you can literally cover the entire Earth every single day. So for example, we get some of our ultraviolet radiation flux data from what used to be the Earth Probe TOM satellites. Now the OMI satellite is going up there. And so you can actually go to these websites and download the new UVB flux from the entire Earth basically every single day. So you've got trade-offs. So the earth science interest at NASA is much more focused on what you can do with remote sensing. And so I've been trying to argue for a year or so that with synthetic biology you have a potential to watermark organisms. So say you have something that might be a pathogen or a genetically engineered organism that you have out in a cornfield or wherever, that you might be able to add a pigment that is not found normally in nature, or certainly not in that organism, and therefore be able to track it with remote sensing. I actually mentioned that the Air Force once, and they said, I think we may already be funding that. So I don't know what's, exactly what's going on. You can imagine that they're using that for different reasons. By the way, that illustration that I've got in the bottom left is from OMI data, so that gives you an idea where when I say there's a total Earth coverage every day, there is a total Earth coverage every day. It's very cool. So that's sort of the idea that I'm playing with in the earth science. So now I've sort of done my pitch for aeronautics and earth science, and so now we can sort of have fun with astrobiology. So apologies for those of you who were here yesterday, but astrobiology focuses on three major sound bites. Where do we come from? And this is evolution writ large, so it's all the way back to the origin of life. How do you get the chemicals for life to originate? How do you end up with a habitable planet? How do you end up with a galaxy? It turns out that you can't actually just be anywhere in the galaxy. There are parts of the galaxy that are going to be too high in radiation and parts that don't have the right metallicity, which to an astronomer, a metal is anything over hydrogen. I mean, it's all completely, I'm saying this with a straight face, so, but it's a completely different sort of thing than you might think about in evolutionary biology and the way it's normally practiced in universities. So it's where do we come from all the way back to the Big Bang. So really bringing in these environmental factors. Where are we going because the Earth is continuing to slow down because of its frictional interaction with the moon. Day length is increasing. My students ask me if that means they'll have longer on the final exam, and no, it's increasing, but very, very slowly. The sun is burning. There are all sorts of things that are happening. So the where are we going is what's going to happen in the future in this astrophysical context. And then, of course, the are we alone? And I'm going to argue that we can use synthetic biology to help answer all three of these questions. So, let's go from there. So the obvious thing is to start with the origin of life. This is, one of the big where do we come from. And what I'll do is, through most of the first part of the talk, really give data from and highlight other people's labs rather than my own, and we'll end up with what we're doing ourselves. So the most basic thing that most of us study has to do with a DNA-based organism. A few people I know study RNA-based viruses, but you're basically looking at a genetic code that is universal on the Earth today, GCAT, and so on. Is there a reason that we have to use those four bases? And yes, I know they're modified bases, the dinoflagellates and so on, but these are basically the four canonical bases. Steve Bennett, you see in this bottom picture in Florida, has for years been playing with the idea of making alternate bases. So what he's created in his students is something that he calls GAT-ZP DNA. So he's added 2 new bases and gotten them incorporated into polymers and so on. So you're starting to play with using synthetic biology. What if? What if we had a different genetic code? What if we had more bases? There was a very interesting paper that came out of the Medical Research Council, Phil Holliger's lab, just a few months ago. Vitor Pinheiro was the first author on that. He's a very talented postdoc. And what they did is go the next step. So it's not just modified DNAs, but actually making modified genetic polymers. So they were able to make these XNAs, they call them xenonucleic acids, and get DNA to transcribe into these XNAs, which it turns out were a bit more stable than DNA. We're able to evolve aptamers, and then we're able to put it back in the form of DNA. And so this is really very cool, very far out stuff, which I think is phenomenal. So this possibility here of playing with, are there alternatives that might occur elsewhere, or are there alternatives that we could use going forward? I mean, you can imagine how useful it would be to, if you were going to say Mars, having an alternate life form that couldn't cross-react with our current life form. It might, increase the safety so you don't get an Andromeda strain sort of thing, or you might have something that's more efficient for whatever environment. So now that I've sort of really given you the sock in your gut over the fact that you could really just start by changing the entire code, you certainly could imagine alternate biochemistries. All of us in this room use L amino acids and D sugars. Why? It's probably some kind of frozen accident. There's no reason to think that's any more efficient than D amino acids and L sugars. And I know Hal Morowitz played a little bit with trying to tweak that several decades ago. But what about starting from scratch and having a whole new organism that uses a different kind of chirality or other sorts of biochemistries at other sorts of levels? Or alternate cellular systems. I think we're going to get to a point that we're not just fiddling around with a few genes, but we're going to dial new membranes and so on. Or even membrane-bound organelles. There was a very cool paper a couple of years ago of someone who had made a hybrid Golgi apparatus. And I mean, this is so incredibly cool. They used a hybrid of microfluidics and proteins. I mean, how cool is that for a Golgi apparatus? I have no idea how they actually were able to do this, but they were able to do that. And so that kind of thing is going to start moving along. And the colleague at Northwestern who looks at, for example, ribosomes and says, look, the main function of a ribosome is as a polymerization machine. So why not take a set, make an orthogonal set of ribosomes that polymerizes something else, like maybe nylon? Why not polymerize something completely different and use that as capabilities of ribosomes? So that's some of what's coming down the pike. Really, completely re-engineering life. So as part of this, there are several groups that are looking for the minimal organism. And there are sort of two ways that people look at for the minimal genome. One is the sort of more origin of life bottom-up approach. So you start with a membrane and you put nucleotides in, and you can add a flagellum if you're so inclined, and so on. So you're seeing what you can do starting from zero. The other approach I call a top-down approach, and this is more taking an existing organism and start pulling out parts and see how far you can go before you lost that capability. So let me give you an example of, or two of each. Jack Shosak's lab, and I know he was here relatively recently, has pioneered some of the work in this bottom-up approach. This is out of one of his papers from 2008, where they made lipid vesicles and they put activated nucleotides in and got some sort of polymerization. Dave Deemer at Santa Cruz has done similar sorts of things. A couple of other labs are working on this kind of thing. In our lab, we're playing a little bit with that idea, but rather than starting at the membrane level, we're starting with the whole idea of re-evolving proteins. So this is an incredibly talented postdoc of mine. I shouldn't even tell you his name because I don't want anyone to steal him away. This is Kosuke Fujishima. And what he has been very interested in doing is, as I said, re-evolving proteins. And what people tend to do is take ones that are already available and tweak. And we're doing a very different approach where we're making these de novo. So let me, he's from Japan, on his 30th birthday. I've been calling, he wore his traditional outfit, I've been calling him Samurai Postdoc ever since then. But basically, the idea in the RNA world, and this is why we got interested in originally, was because of the origin of life. And you have this sort of canonical idea that you start with a prebiotic world, where you go to an RNA world. The idea is that RNA can replicate itself, so it's got hereditary information, and it's also got some catalytic capabilities, so it can be both the genotype and phenotype. And then at some point you add proteins in, and then at some point you have a protein RNA life form, and eventually a DNA coup, I call it, because DNA is more stable than RNA. And what we're saying is actually you don't ever have an RNA world. And there are a couple of reasons for this, that nucleotides are much more complex than amino acids, and I'll show you a little bit more about that in a moment. Self-replication of these ribozymes is actually still problematic, and RNA is unstable in aqueous solutions in the presence of things like magnesium. And so what you'll see in a moment is that we're proposing an alternate route for the origin of life, that in fact you start with a peptide world, and you go to a peptide RNA world to a DNA world. So in fact, you run the central dogma backwards. Now, part of the argument for this is that if you look at sources, abiotic sources of compounds, you find out there in the interstellar medium and so on, amino acids. You also find PAHs and so on, but what you don't find is DNA or RNA. So here's a great paper that I find very inspiring from Paul Higgs and Ralph Pedruzza up at McMaster that they published a couple of years ago, and I've highlighted bits so that it makes it easier for you to see. This is the relative abundance of amino acids, and if you look at the meteorites, stardust, ice analogs, prebiotic experiments, hydrothermal syntheses, other chemical syntheses, so in other words, all abiotic ways at making amino acids, what you see is a pecking order of amino acids. There are about 4 to 6 that are relatively common, 10 that are out there, and by the time you get towards the end of this list of 20, you don't even see them. So there is very definitely a pecking order in the ease of synthesis and the thermodynamic stability. And so what Koski and I decided is, let's start with the ones that you would have found on prebiotic Earth, this first 4 to 6 really stable, really commonly found amino acids, and see what kind of capabilities you can make from there. He's also now sort of taking this next step to 10. And so what he did is he designed sequences of DNA that would code for structural motifs like alpha helices and beta sheets and so on, but only using GADV. And then randomize that, which is what he did last summer, and now is at the point where he's starting to express them using a pure system and expression methods so that you can actually go through cycles. So what we're trying to do now is see what kind of capabilities we can evolve using just these 4 amino acids. He's now moving on beyond the fantastic 4, and you can see you very quickly get thousands of variations. And so what we're doing now is just, when I say we, of course, I'm here, he's there working, is doing all the basic sort of things that you would want to do on a prebiotic Earth. You would want to start to well, these aren't actually in the order I would put them, but peptide bonding, peptide transfer, proteases, and then you might want to start making nucleotides and polymerizing them and phosphate transfer and all sorts of really basic capabilities. So it'd be really interesting. So we're doing a really extreme bottom-up approach, not at the nucleic acid level, but at the protein level. And that's where he is so far. Thank you. Now, an opposite approach, as I mentioned, was doing sort of a top-down. And the really common organism to start with is Mycoplasma genitalium, which is a parasite. So it already has got a very lean, mean, reduced genome. And this is a figure out of John Glass's work and colleagues at the J. Craig Venter Institute on the East Coast, the Rockville, Maryland part. And what they did is they took this mycoplasma and they used transpositional mutagenesis to see how many genes they could knock out and still have it functional. So it started with 482 protein-coding genes, and they were able to get down to 382. You think about that, I mean, granted it's a parasite and so on, but still, that you could function with only 382 protein-coding genes is pretty remarkable. So we're starting by going both directions and getting at what the minimum size might be for a cell. But these are techniques that help to inform what life might have been like on the early Earth or anywhere else in the universe. But doing it, taking advantage of these tools of synthetic biology. This is just sort of an aside. I now have a group of engineering students, primarily and some biology students working on 3D cellular printing. So, you know, what about digitizing a multicellular organism the way we digitize nucleic acids or amino acids, take them apart and then reprint them. So we've just gotten a really specific microdispensing system, positioning system in just the other day, so we'll be starting to do that. So I know I spoke to someone here in the engineering department this morning who's doing some really nice 3D printing with plastics, and theirs goes down to, what it was, 30 to 50 microns, I think. But if you're starting to deal with cells, even eukaryotes, even something like yeast, you're going to have to get down to the 5 to 10 micron range. So that's the kind of thing we're playing with. And there's Sam with his hand in a sponge tank, perfectly nice engineering student now worrying about raising sponges and so on. Sponges are kind of obvious for a first attempt at a replicator because it's going to work. Anyway, I'll explain why later, but basically there have been deaggregation studies done since 1909. Those were not actually my studies in 1909 for the younger people who might be confused. But it does actually work. They do reaggregate, so there's no reason we shouldn't be able to print them back together. Where do we come from? I think some of you saw this yesterday. There are a lot of extreme environments out there. And to get an idea of what the envelope for life might be like in the universe, we get an idea by studying life in extreme environments on the Earth. So there's this enormous number there on the Earth, but if we want to expand that a little bit, why not make an artificial extremophile rather than waiting to go to planet X or Y or moon Z and finding something that pushes these bounds. And so this second postdoc in my lab, Ivan Polina Lima, who's there about to lead a bunch of us into caves in Brazil, has been looking a little bit at one aspect of making an artificial extremophile, and that is focusing on this whole idea of Michael Daly's that manganese is a key to radiation resistance. So whereas Michael Daly's been doing the biochemistry, very nice work over a decade or so now, showing a link between manganese and content and resistance to radiation, we're taking the opposite approach of going out into the field to high manganese environments and seeing if we can get something that knocks Deinococcus off its pedestal. So here's some of his initial results. Unfortunately, the two slides I show you are color-coded differently. But what I tried to do right before I started talking is helpfully add a few arrows. So what you see there in the red is E. coli, which is, of course, pretty lousy at radiation resistance. And the black arrows point to the Deinococcus radiodurans kill curves. And the middle ones are isolates of his from high manganese sites. You see a manganese mine. NASA, it's slightly embarrassing, but we for many years combined NASA with a Navy base, Moffett Field, and so I did have it in my mind, and I was right that we at one point possibly still are a Superfund site, which is pretty lousy, but it did occur to me that since if we were a Superfund site, there might be some high manganese right there, and so that it is. So this is not sort of like a typical, I would assume that your universities do not have manganese levels like that, but we did. So you can see we're already getting pretty close to something that's the level of Deinococcus radiodurans. Here's a sample and an isolate from the Atacama Desert in Chile. And say, unfortunately, the color coding is a little bit different. So here's that black arrow again for the Deinococcus radiodurans, and then there's E. coli not making it anywhere. And this time you can see on that light blue line that he's actually already found an isolate that is more resistant than Deinococcus radiodurans, is Hemenibacter. What's kind of interesting is it looks like it probably also has a virus. And so it's not clear to me whether the virus is perhaps helping in the radiation resistance, or without it, it would be even more radiation resistant, how this is all working. And so we're in touch with some virology labs right now and working on it. Yvonne, I believe, right now is on a plane heading for Brazil, and we'll be doing another field trip down there in the next few weeks, at least that's what he told me. So what we've done is very quickly, by going to these high manganese environments, started to find organisms that are more resistant to radiation than even Deinococcus. And the next step is clearly to take some of these genes from these organisms and put them in other organisms to see if we can increase radiation resistance. And that's kind of a spoiler alert, because I'll give you some data from that later. There's been a lot of interest in astrobiology. I've been interested in trying to derive general principles. So we have this narrative of the evolution of life on planet Earth now, where you have the cooling of the Earth, and you have the origin of life, and at some point you have, cyanobacteria and eukaryotes and this and that and the other thing. But that's a single narrative. And so what if you replayed this table of life? And this is something that I know, Steve Gould over the years has talked about and argued for contingency, and Simon Conway Morris is also a friend of mine, as Steve, so I'm not sort of trying to pick camps here, was really pushing for a certain necessity for certain pathways. But the fact is that you're somewhere in between, that there's a certain amount of contingency and a certain amount of predictability in evolution. So I've already sort of given away part of the story for amino acids, that you can even predict which amino acids you're going to start with. But this is the kind of thing that is amenable to looking at with synthetic biology. You can do games like try to hypothesize universal common ancestors of particular genes and then play around with them, change amino acids and proteins and active sites, compensatory mutations, and so on. And so it's not just waiting for nature to have come up with a couple of varieties that you can compare, but you can go into the lab and actually do that kind of thing. And did the common ancestral gene function better at high temperature, low temperature? This is kind of the work that Eric Gaucher's done at Georgia Tech and so on, which I find absolutely fascinating. So that's sort of the looking past astrobiology angle. Of course, in the future, we're going to use synthetic biology to do all sorts of things like pharmaceuticals and so on. And since the future is fair game for astrobiology, I feel justified in being able to put in a slide like this. But the fact is that we're not going to be able to live on planet Earth forever. And so this where are we going bit to astrobiology also includes going off planet Earth. Now I quickly point out this is not a photograph. It's not like I have these hidden, aliens. I actually pitched this to Charlie Bolden, our administrator, a year ago, and he sort of looked at it, scans and said, no, it's not a photograph. This is what's going to happen when I show up on Mars. So human exploration is actually a rather interesting sort of venture because you have all sorts of needs for human settlements. Transportation, habitats, life support. When I talk to the military guys, I say, well, it's really hard sending troops to the hills in Afghanistan. I say, you think that's bad? At least you've got oxygen and gravity. We don't even know that. So, it's really the ultimate field trip to go to some place like Mars or the Moon or whatever, and you need power, heat, light, and so on. There are a couple of issues, though, and they mostly boil down to upmass. And what I mean there is just lifting something into space. The number that I've heard tossed around, so I repeat, is that it costs about $10,000 to launch a Coke can into low Earth orbit. Okay, so picture that. $10,000 for a Coke can into low Earth orbit. You're not even at the Moon yet, or Mars, or anything else. This is very expensive, so any way that you can reduce the upmass, you save enormous cost. So you can see where I'm heading here. Something that's living that could then reproduce once it's somewhere else could save you a huge amount of upmass. You also have issues with storage and flexibility. So I'm involved right now in a mission that we're hoping goes on a German satellite in 2015. And again, as a lab-based scientist, you're even doing field work, trying to get something back from Kenya over a couple of days seems like a big deal. For these space flights, they say you have to be prepared to have your sample sit on the runway for 3 months. There could be 3 months of delays. I mean, that's a nightmare when you're trying to do biology. Oh yeah, and by the way, it's got to be able to sit there for 3 months, untended, sealed in a rocket. So storage issues are there. And again, you can imagine using biology, particularly as we've gotten so much better at cryopreservation, plus organisms with spore forms and so on, there could be a huge advantage there. Plus flexibility. This may come as quite a shock to some of the younger members of the audience, but Amazon does not deliver on the Moon or Mars. You better have it with you and be able to rejigger as needed. So if an astronaut's up there and gets sick and you didn't happen to bring the right drug, oh dear. So it would be a whole lot better if you had some flexibility in manufacturing. And so the argument is that all of these are areas of synthetic biology can be enormously helpful. So I pitched all this about a year and a half ago to a group of they really were aeronautics people at the NRC Council. And I thought that I had blown their minds away. And they look at me in their ties and white shirts and said, so, what's the big idea? I thought, whoa, wait a minute, I just gave you all my big ideas. But you can't say that, so I took a deep breath and I said, all right, you want the big idea? Star Trek. So, let me give you some of these sort of really far out but not so unbelievable ideas. For millennia, we've been using biology to do chemistry for us. Obvious, you know, beer and wine are obvious sorts of things, but in fact, we use biology to make silk and wool and cotton and wood and you know, enormous number of things that we use and wear and eat and so on. Is there any reason that we shouldn't continue to use biology just because it's planet Earth? We could get material from the Earth, but then you've got the mass problem, or you could acquire materials such as water and organic carbon, or even CO2 and nitrogen gas when you've arrived at a place, or maybe, say, in transit from an asteroid or something, and use this to make products such as foods, nutritional supplements, gases, clothes, and so on. You might use particular microbes for on-demand biomining to pick up metals that are just simply too heavy to take with you, or for habitat construction. I'm going to talk a little bit more about that in a moment. Or this whole idea of the alternate biochemistries, which could be actually safer or conceivably more useful for us elsewhere. So let's start with this whole habitat idea. There have been sort of crazy ideas that we've thrown around about doing things like making a spider silk scaffold, because they've certainly already engineered microbes to make spider silk, and then maybe cement that to have some kind of calcite precipitation. But we've gotten rather taken the last year or so with this idea of biocementation, where you take organisms and you use them to precipitate calcite and make a brick. So this is something that my iGEM team worked on last summer, and I'll show you some of their slides in a few moments. The biomining is things like taking these metals out of the regolith, that's the surface of Mars, and I have this graduate student, Jessica Navarrete, starting on that, and she is specifically focusing on urban biomining. Again, I'll show you the iGEM students project with regular biomining in a few moments. But the idea of urban biomining is recycling metals that are already in electronics or parts of spacecraft. You know, once you get to the moon, like remember the Apollo missions, we left the landers there. Why not take some of those materials and recycle them? You've already spent all that money getting them there. If you're using these big metals the way they are, they may not be digestible, but you may be able to put them in a form where they are could be released. And in fact, Jessica is specifically starting with electronics. And I either dread or can't wait till the first paper that we publish that has a materials and methods section of, you know, grind up 2 iPod Nanos, make 10 millimolar solution of same, you know. But there's going to come a time that Jessica is going to have to be, you know, stripped down and searched before she enters any of our offices, because she will be very dangerous. So I won't tell you how exactly she's going about that, but I think it's a very cool idea. So that's some of the stuff going on. So now I want to turn it over to where we've gone with those projects. And so let me give a quick introduction of the iGEM program. I know some of you are familiar with that, and we even have one alum of the iGEM program. What this is the International Genetically Engineered Machine Competition. And do we have anyone on from the University of Washington? Okay, they won the whole thing last year, so they know they're very familiar with that. What this did, it started off as a competitive molecular biology project. Well, actually, it started off not competitive as a class at MIT, and quickly became a competitive project with several other classes in the Cambridge area, and then very rapidly expanded. So it started about 8 years ago, and this year we just had the world's last weekend, and there are 190 teams worldwide. So these are teams of primarily undergraduates. Some of them are as tiny as 4 students. Some of them are as massively large as 25. The story is the University of Washington also has a feeder team of another 25, so they basically have 50 students working on it. But most of them are in the range of about 10 to 15 students. So what happened is I had this reconnection with Brown. I also teach astrobiology and space exploration at Stanford. And for various reasons, neither school was going to have a team last year, and for some reason I was sort of in the wrong place at the wrong time, or the right place at the right time, and ended up putting together both universities and pulling students from both. So this was my team last year, and what you see up there are 3 students from Stanford University. and what is it, at this point, 6 students from Brown University. There's a slight change after a while. And this is right after they gave a really stunning presentation at the regionals in Indianapolis, because at this point it's gotten too big for a single world. And I'll just tell you that I'm very proud of them. They won Best Presentation, as 63 teams went to the Worlds and won Best New Application in top 16. That was only 160 teams last year. But still, I'm extremely proud of them, and it was sort of like one of those late night, TV movies with the scrappy little high school basketball team that can't even dribble, and then they go on to win the state championship. Anyway, that's how I felt about them. They were terrific. So they were the first really to take some of these ideas and come into my lab and to start to do them. And part of the conceit of the iGEM competition is that you can take these standardized genetic elements, what they call RegoBricks, that are all deposited at MIT, and the students take and then make new ones during the course of summer. They have standardized cut sets at either end, and so it should be a little bit easier to engineer new pathways, do novel things, and some of these teams have really done spectacular things. So they have the summer to do that, plus some human practices, some outreach, some ethical consideration, and then they have to do a 20-minute presentation, a wiki, and a poster, and then at this point the top teams go on to the Worlds and repeat the whole thing. So all of these things I'm going to tell you are still online. You just Google for the last year Brown-Stanford iGEM 2011, and you can find their wiki, and it goes into much greater detail where you can actually watch their 20-minute presentation. But I'll just give you sort of the highlights. So they were the first to really look at this issue of using synthetic biology to have a human colony. And they focused on 2 major things. One they called power cell, and one was the biocementation. So the idea of power cell is, say you're going to go to Mars, or the moon, or someplace, and you're going to make drugs and fuel and so on using Bacillus subtilis or E. coli or yeast or something like that. You're not going to be able to make Chateau Mars without organic carbon. And at this point, besides methane, we've yet to find organic carbon on the moon or Mars. So what their idea was to do a co-culture, starting with the diisotrophic cyanobacterium. They ended up focusing on Anabaena, which is a filamentous one. I'll show you in a moment. Diisotrophic means that it fixes nitrogen as well as carbon dioxide. And then engineered that to secrete sucrose. It already releases the fixed nitrogen, and then use that to feed, in this case, an E. coli. It turns out that was kind of tricky. Here's the Anabaena, and the nitrogen comes in, and you get the nitrogenous products going from this heterocyst, this specialized cell that doesn't really do much photosynthesis. It goes out to the vegetative cells that are more photosynthetic. There's one of their micrographs in the background. And so what they focused on was increasing the sugar production out of this Anabaena and having it secreted into the medium. And so they engineered the CSB sucrose permease and increased the production in addition by giving it some salt stress. So it was there as an osmoticum. So sort of to cut to the chase, I don't think I showed how it was done. They were very successful. It turns out that it's not an easy thing to do because Anabaena doesn't like to get exogenous DNA in it. It's got all sorts of restriction enzymes that basically will cut up anything that's put in. So what you end up doing is having what's called a triparental mating. So you engineer into one plasmid, you put in an E. coli with a helper plasmid and a methyltransferase so that you really are camouflaging that DNA. You put the E. coli and the Anabaena together in a test tube, you turn the lights down for the night, you play soft music, and in the morning, if you're really lucky, they've done their thing. And so what you can see here on the left is just a light micrograph, and I'm not quite sure why, but it looks like the ones that were successful in transformation are a little bit lighter green than the others, so there's some sort of stress, obviously, on the chlorophyll. On the right, you see under fluorescence what a very successful transformation. So what you see is this string of green cells. These are the vegetative cells that now have the GFP marker on the sucrose permease, so we know it's in there. And then every now and again you see a gap, and those are those heterocysts I was telling you about that primarily fix nitrogen. You'd see a little background fluorescence because there's a tiny bit of photosynthesis going on. But basically they were very, very successful doing this in the end. And the postscript on that one is it looks like that's the experiment we're going to be flying on this German satellite. which is very cool because the idea that particular mission is flying under 3 different gravity regimes during the course of a year and a half. So you get, basically 0G, a 6th gravity, which would be more like the moon, and a 3rd, which would be more like Mars. So we're really excited to see that power cell then move that next step into space. So that's why we're now starting to move on to these issues of stability on the launch pad, and instead of using E. coli, use bacillus because it's going to be more stable and so on. And that's actually where the euglena and the clammies is coming in, because we're going to try that in some of the wells as well. Their second big project was this whole idea of building habitats, and so this is something that they called RegoBricks. And as I said, there are people who have started to play with using Sporus pasteurii and mixing it with anything from sand to glass or whatever, and adding some calcium carbonate, and then having the cells, and some urea, and having the cells then increase the pH in the medium. The calcite is precipitated, and you end up with a brick. So the brick that you see in the upper right there is not one that we made in our lab, but it's been made with this sort of technique. And there you see a photograph of the Sporus pasteurii. So the idea was to take this urease cassette, which it turns out is very large, and put it in E. coli to start to mix and match. I've mentioned this to a couple of you the last day or two. Going to the synthetic biology is complete reversal for someone who's used to evolutionary biology and celebrating this diversity of life. You're suddenly not looking at a diversity of life, you're looking at life as this giant hardware store of genetic parts. And you now can do this sort of swapping and moving around. So that's exactly what they did. They were able ultimately to get the urease operon out of Sporus pasteurii, put it into E. coli as a proof of concept, have it produce the urease gene, and therefore make ammonia out of urea. And obviously, if there's a human mission, you've got plenty of urea, and therefore be able to precipitate the calcite. Now, it turns out there are other sorts of issues with using that approach, and I really would rather not send E. coli to Mars because they're not even 90-pound weaklings. They're like 90, milligram weaklings or something. Something you want a tough guy in. Bacillus subtilis spores have actually been flown in space in low Earth orbit for I think it was 6 months and survived fine. So these guys are already flight tested. They're great little astronauts, and so we'll probably be moving more towards that. Sort of the postscript on this is that NASA headquarters has picked it up for a year, and we're starting to move into sort of a multifaceted approach where we don't rely just on biocementation, but also bioglues. So my colleague, David Loftus, is an MD, PhD, has been using some human glues to then start to cement together Mars simulant. We're starting to take glues from nature, from the intertidal and so on, and do it that way. So I think it's going to be a really, really interesting project, and that's one that's gotten a lot of press attention. But a lot of this won't do any good if we won't be able to get it to work on Mars or the moon or wherever. And so, because our lab has done a lot of high-altitude ballooning the last 5 or 6 years, we basically, for almost no money, a couple of tanks of helium can get you up to about 33 kilometers, which is through a lot of the stratospheric ozone layer. So I don't know if you can see on that, figure that I made up there, but once you get up to about 33 kilometers, you can see you've gone through about 90% of the ozone layer. You've lost a lot of your atmospheric pressure. You're now down from about 1 atmosphere to about 7 millibars, which is roughly what it is on the surface of Mars. And the temperatures, you go through a couple of different cycles there. So you're at least picking up basically the solar radiation that's not exactly like Mars, but closer than what you see on the Earth. You're going through these extremes in temperature, you're going down to that pressure. And we didn't take up genetically engineered organisms because obviously that takes a lot more permitting than we were able to do in a fairly short time. But we did take the hosts up and look for survival and showed survival. And that's one of the actually flight curves. So if you're familiar with the Bay Area, you can see the Pacific Ocean out there in the blue, and there's the San Francisco Bay. We were on the east part of the bay. So if you go straight through with that highest part, where the balloon burst and go to the ocean, you pretty much go right through Palo Alto, through where Stanford and NASA are basically located. So that was very cool. And if you've never flown something on even a balloon, it's a little bit home movie-ish, but it is so cool to take your experiment, put it in there, and just watch it go up. And that particular night, we had a film crew from the BBC, and they didn't like where we were launching. It wasn't photogenic enough, so they found another site, and we set everything up, and apparently we wandered into a different county that we didn't have permission for, so we were chased off the property, and we were sent back, and on and on. So it ended up bursting at sunset, and it was amazing. It almost looked like it was Venus up there, and then all of a sudden, it was gone. But the result was that we didn't end up recovering it till the next day. We were tramping around a gated community at midnight that apparently had some rattlesnakes. You wonder how I ever get any students in the lab. But they had a great time. The problem is the BBC didn't like the fact that we didn't have it in hand when they had to leave and say Eureka. So in the end, we weren't in the American version of that special. But it's a very cool kind of approach. So anyway, that's what these scrappy, really very bright 9 kids did last summer with me. Sort of the blind leading the blind, but they did a phenomenal job. And no good deed goes unpunished, so the word got out that we were having an iGEM team, and I got 50 applicants this summer and said this was iGEM Stanford Brown 2012, Mach 2. You see it's a much younger group, very excitable. So we had this time 5 undergraduates from Brown University, and 8 of them from Stanford, and they did 3 projects, but they were focused much more on astrobiology, and so that's why I wanted to really end with them, because of the transit of Venus this last summer. I'm sure you remember that. We ended up calling it the transit of synthetic astrobiology. And what I'm showing you is just a very tiny portion of what the amazing thing they did was. And what's so wonderful about students who are at that level is they don't know the meaning of the word no, and they don't know the meaning of the sentence, you can't do that, it's impossible. And so they really did a couple of PhD theses during the summer that I did say periodically, you're biting off a lot, because I knew no would just be taken as a challenge. But the amazing thing is they actually accomplished basically everything they set out to do. So they had 3 basic projects. One, it was HellCell, which is basically making artificial extremophiles. So I told you a little bit earlier about Yvonne's work with the manganese transporter. They, of course, are not satisfied with just radiation, so they wanted to look at desiccation resistance, radiation, pH, heat, and cold. Used all of these genes, biobricked a lot of them, put them into E. coli, and unfortunately, the graphs, that's what I was doing up here, that show all the results didn't seem to come through with the transfer from PowerPoint and the various versions and fonts and so on. So I'm afraid you're going to have to take my word for it. But every single one of them ended up better than the E. coli control. I mean, it was really remarkable. In some cases, like with this manganese transporter, substantially more resistant to radiation. So, I mean, it was really stunning to me that in the course of nominally 10 weeks, it really was more like 3 months, that you could get, there were about 5 students working on this part, 4 or 5, that you could actually produce 5 different types of extremophiles, and then they did several within each. And so I think as a proof of concept, that was quite stunning. Obviously mixing and matching would be a very interesting next step. They looked at the biomining, and this was something that is going to be very important, I think, for our future, both on the Earth and off the Earth. Many of you probably don't realize that something like 25% of the copper mined on the Earth today is already biomined. So we can go on from there. And I mentioned the urban biomining. They focused more on trying to take materials out of, for example, the regolith. And the idea at the beginning was that you could recycle and utilize materials in space. You would get around the bulky, hefty, expensive traditional machinery and using organisms that would be cheap, light, replicable, and non-toxic, which they didn't mention, but it's true. So their idea at the beginning was to actually link binding proteins, this proof of concept was copper, to the cell surface. I guess someone had done a little bit of that before, and so I said to them, you know, basically it seems to me what you're trying to do is increase surface area to volume ratio. You ideally don't want to put the metals inside the cells because many of them can be toxic at high concentrations. You want to keep them outside. So I said, you know, the ultimate surface area to volume ratio appendage on an E. coli is the flagellum. So they went into the literature and they found a particular gene, this FLIC gene, and put a GFP marker in it and the multiple cloning site, and ended up then inserting, and that's just showing that the GFP was expressed in the flagellum. And again, I'm sorry, I don't have the other images, but they ended up adding a copper binding protein. Doing bulk assays, and were able to show that in fact they were binding copper in an exterior way because they get a decrease in copper when they spin down the cells. And in addition, they're starting to do the sort of SEM, mineral analysis, EDS, and so on that you really need. But that they just started doing that last Wednesday night as they were getting on the plane for MIT. So I don't really have good data to show you. We don't really have anything positive at this point, except some hints of some copper peaks. But that's obviously going to be the final proof positive step. But it looks like we've now set up a system that could be used for biomining by switching out different metal binding proteins into this flagellum. And finally, this is kind of the most fun one, so that's why I left it to the very end. The question has been for a while, as I mentioned yesterday in the talk, whether organisms could live in the clouds above Venus. Now, the idea, sort of to refresh your memory, is that Venus should have been similar to the early Earth, oceans and so on, because they would have gotten the cometary flux and so on like the Earth and Mars. But what happened is, being closer to the sun, they ended up with a runaway greenhouse. And so the oceans went into the atmosphere, the surface became extremely hot. It's the hottest planetary surface in our solar system today. But there are places in the atmosphere with a large amount of water vapor, the atmospheric pressure of the Earth, and about the temperature that would be certainly pleasant enough for life, clearly well within the 0 to 100 degrees Celsius range. The problem is, if you look up, we don't have green and pink and orange clouds on the Earth. And we know there's stuff living on the Earth. We know there are things in the clouds. That's been known for decades. And yet we don't know if any organism can actually complete a life cycle in the clouds. So this is something that's been bothering me for a while. And so that was part of what we were doing with the ballooning, was trying to do air capture. And I was thinking, well, you know, you could do ribosome to DNA ratios to try to get an idea if they're dividing. You could do all sorts of things. You could try to catch them, you know, morphologically looking like they're dividing. But every time I thought of something, you could sort of talk yourself out of it and say, well, maybe they were just basically flash frozen as they came up off the surface, and it doesn't mean anything. And so it finally occurred to us that we could use synthetic biology to get at this question that's been, you know, around for a while. Is it conceivable that life could live in the clouds above Venus? And so part of it was one student did some modeling about the retention time in the atmosphere. But what the other students did was something that was very clever. This is just telling you how really nasty Venus is. Sorry, I sort of jumped over and missing all these wonderful animations that Jason, who was on the team, did. So let me cut to the molecular biology. I apologize for that. They used two approaches, and one was basically to take a GFP and make a construct with the ribonucleotide reductase gene. And so you don't produce deoxynucleotides from ribonucleotides unless you're replicating the DNA. And the other was Pol A. So basically what they were doing was creating E. coli that told us when they were dividing, rather than us just trying to count cells. Now the other part, they were actually phenomenally successful, and I don't know, well, here's just showing some of the, you know, that it certainly, the transformations worked. They did nice bulk assays where you could see the periodicity once you cell synchronized with the Pol and the NRD reductase. They actually got best new part at the America's West regionals for this NRD gene construct. So the whole idea was to then take that and put it in an aerosol, and that's where we had problems. we're trying to modify a milk and oil drop apparatus to get them suspended, and this was what one of my engineering students was working on, and there were all sorts of technical difficulties with that. The great news is that just 2 weeks ago, we presented this at the Space Grant Directors Meeting in Seattle, and afterwards, one of the people involved with that at Johnson Space Center came up to me and said, would your students like to fly on space stations? Like, we've discussed this every day since June 1st, absolutely. So we're very much hoping that we now can take these constructs, or a parallel one with in bacillus or with a bioluminescent marker, and put them in a nanorack on ISS, because once you're in space station, you no longer have to worry about gravity with these aerosols, and be able to really finally answer this question whether it is possible for even Earth organisms to glide while they're in an aerosol. So that's the team this year after the America's West. So that was sort of giving you a huge idea of the sort of things going on. I do have some colleagues who are starting to look at what you could do with food preservation, life support systems. They call it ECLIS, environmental controlled life support systems using synthetic biology. Obviously, much of our life support on the Earth today is because of biological life support. NASA does not use biological life support. At this point, it's all engineering answers, and so if we could start to bring biology into space, it has all these huge advantages. But other than that, it's a very tiny team of us right now. It's something that I've argued that, unlike most other NASA projects, we don't have to take the lead, because there are lots of other people doing synthetic biology all over the world. We can leverage off that investment. We can contribute to the field by the fact that we are at the forefront and work on life in extreme environments and origin of life work. We can also push the field because we really need this miniaturization and automation, because we need to go into space and we have the upmass issue. Most people don't care. You can go into a lab, and I'm sure many of you have labs, where you've got a piece of equipment the size of this table that weighs 300 pounds. It's only painful the day you have to move it in or the day you have to move it out, but it doesn't bother you while it's there. It bothers us because of the upmass issue. And so we can help drive the field towards miniaturization and automation, which will ultimately be useful not just in space exploration for planet Earth as well. So those are sort of that's where I see our role at NASA interfacing with synthetic biology. And so in spite of the fact that this was sort of thrust on me, I have actually become a great believer that it's time for NASA to join this synthetic biology superhighway. Thank you very much. I don't know if I'm supposed to take questions or comments or rotten tomatoes. Or
I have a couple questions.
Yeah.
So one question about your four-letter alphabet where you're trying to build protein secondary structure.
Yep.
Another question about your biomining and biomanufacturing in general. So my first question is sort of like, just in general when you're designing protein folds, just like secondary structures, like how easy is it to like get it full and not this full, like in terms of percentages? I think my second question is, if you like look at like a mining operation, you go through like, you know, massive quantities of rock very rapidly with these giant machines. And given like the biological constraints of like having, you know, biological standard conditions, you know, temperature, pressure, etc., I'm wondering whether you can ever have enough material flux through any sort of biological system for it to ever be cost effective. Because you can't have like, you know, a bunch of microbes at 500 degrees Celsius, you know, running lots of stuff.
Right, right. So the first one is on the folding, and I don't think that there's been any problem. But I would direct you to talk to Koski about it, because you're dealing with, you know, a very tiny alphabet, and so there's not a whole lot, you know, a lot of leeway there. In terms of the biomining, there's sort of two answers. One is if you go to the urban biomining where you're recycling what's already there, you know already what's there. And so it becomes much easier because it's already concentrated. So part of the problem is things like, you know, silica glass that surrounds some of the electronics, some of the wires, and getting that away and going in. So that's actually where we're starting. So you know that. In terms of out, you know, mining from a regolith or whatever, I mean, yeah, you could try to go for a vein. I was talking to Jim Head the other day. It's not clear whether there are any major copper deposits on Mars. I don't think we really looked for it. But the point of using biology is you don't need to go up to 500 degrees. And you may have a long time. I think that one of the suggestions, which I think is a really good idea, is if you're going to have a human mission, you're not supposed to say manned anymore, human mission to Mars, you want to actually send out supplies in advance. You don't want to take everything with you. So it would be great to have a structure there to go to. You know, could you imagine my horror if I'd gotten to Lansing the other night and discovered that I had to build my own hotel room? You know, you want to have the infrastructure there, and that would also give you quite a bit of time then to start things like biomine and so on. But you're right, probably the time scales would not be good enough to really rely on, you know, sort of instant. But that's, you know, somewhat true on the Earth too. Silence, yes.
So when you're talking about things like using cyanobacteria to fix carbon and nitrogen for space missions, it always makes me think of terraforming.
Mm-hmm.
Right? I'm kind of wondering how realistic the possibility is to do something like that on a grand scale on the planet. Has anybody thought of it?
Well, cyanobacteria terraform the Earth, you know.
Sure, right.
There are other organisms. Just
well,
quickly, terraforming means making it like the Earth. Ecopoiesis is just making it sort of habitable. So part of the problem is that Mars lost most of its atmosphere because it's small, so the tectonic activity ran down, it didn't have the gravitation, and so on. So I understand there is enough CO2 in the polar region, and people suggested this, that you could bomb Mars, put the CO2 basically back into the atmosphere, so then you'd have the greenhouse effect, and therefore you could terraform it. The problem is Mars isn't going to be any bigger, and so you're still going to ultimately lose it again after 1 million years, and you've just gotten rid of your entire inventory. So I have actually heard people suggest putting it in a bag. When I say hit Mars, I have a few issues with bagging large terrestrial planets. I think there could be some technical difficulties, among others. So I suspect that what's much more likely is that you're going to have structures. And I think that was part of the idea of Biosphere 2 in Arizona, is to see what would happen if you have a large structure, you know, sort of a large enclosed ecosystem. So I think that that's probably much more realistic. So in an enclosure you could terraform, but to do the whole planet, unless you're willing to say, okay, is this going to be a 1-million-year experiment, and then that's it for the CO2, it's going to be very difficult. Yes?
So you mentioned the issue of clouds on Earth not being green or red or having any signs of bacteria.
No, they do have bacteria, but not a high concentration.
Life cycle. Could that just be because of a temperature issue on Earth that you wouldn't have on Venus?
That's certainly possible, because the clouds here are much colder. The other question would be persistence, whereas they're persistent on Venus because it's basically covered. I tried to use that as an excuse to do field work in the Amazon, but I'm not getting much traction. So yeah, those certainly are two possibilities. But you know, I know there are a lot of microbiologists in the room. If you take or people who bake bread, if you take a dried yeast, it doesn't take very long to get it back up and running. And so you would think that if that were the only problem, that they would very quickly, as soon as there's some condensation, reasonable temperature, that they would be moving very quickly if that's really how they're, you know, focusing their life. And that doesn't seem to be the case. You find, by the way, fungal spores and bacterial spores, primarily bacillus and so on. And by the way, clouds are much lower, as you can imagine, than where we're going in the balloons. Well, you all know, going up in an airplane, clouds are reasonably low, so you're not really at that same sort of temperature inversion and, you know, UV flux and so on. It's not a totally unreasonable place to have a life.
Can anybody see if people at the partner school have questions?
Or going home. Fair enough.
Some people in Texas. I don't know if they're their hands up.
Can you see if they have?
It's a big state. There must be some people in Texas.
I don't see.
What I would suggest, if people don't have questions, because I know this is sort of a different way of thinking about a lot of this, but as you can see, we're a very tiny group that, I said yesterday, I would encourage you to email me if you have ideas for collaboration, or for us to do, or for you to do, you know, or whatever. But I think that there's a lot of really exciting things that can be done when you start to think about using synthetic biology for astrobiology, for space exploration, certainly evolutionary biology. And I tried to indicate I'm not the only one doing this, but I'm one of the only ones within NASA doing this kind of thing. Okay, thanks.